Brody is the Chief and Senior Investigator of the Genome Technology Branch at the National Human Genome Research Institute. I met him when I did a postdoc there before starting as a faculty member here, and had the opportunity to work with him on the multiplex initiative, which I'm really looking forward to hearing about. I haven't had much contact with it for the last 4 years, and I know that it's morphed into something totally different than what I was working on 4 years ago. Dr. Brody, like many over at NIH, is an overachiever. He also runs a lab in Baltimore, has 3 young children, and if that weren't enough, he was just telling me about the menagerie of animals in his home, including the squirrel that they are nursing back to health. So good luck to the squirrel. I have a couple of parting gifts, which I will give to Dr. Brody, but I want to tell just a quick story before introducing him to give you a sense of his personality. So I'm going to put him on the spot here. He has no idea what I'm going to say. When at NHGRI, and in fact at all of NIH, we have to go through a recertification process every year in human subjects research protections. And so he and I were in the same class as we recertified, and he got a gold star for answering a question correctly. And so he had carried this gold star around on his hand for most of the day. And the next day, I was in my office, and I had a coin from Canada that was just happened to be laying on my desk. I think I had given a talk in Montreal, and the little gold coin had this gold sticker stuck to it. And it completely threw me. I could not figure out where in the world this gold star had come from, and so I thought back over a day who had been in my office that could have possibly left this coin with the gold star. So he doesn't know it, but I, in fact, have saved the gold coin from Montreal. So you have $1, Canadian dollar, with a gold coin, with a gold star on it, as my good luck piece that I'd now like to give back to Dr. Brody. And now you're going to tell me, a lot more now, too. And now you're going to tell me you had nothing to do with it, right? I also have a Bioethics Student Association T-shirt for him, which I'm sure he will proudly wear at NIH. A funky DNA-type magnet. He works for the government. He's not allowed to accept the honorarium, and he's not allowed to accept anything of real value. So I had to be creative here. Let me just show you the T-shirt, so everybody's going to want one. So front, Bioethics Student Association. Back. So if you want one of these, please feel free to join us, and I think we have more. And then last but not least, I brought him a nugget of iron pyrite in honor of this talk. So Dr. Brody, the podium is yours. Thank you.
Thank you very much for the nice pass around the iron pyrite. I'm always wondering whether people know what iron pyrite is. You'll probably get it back because I think most people know it is not gold, but fool's gold. And is the mic picking up for you? Okay, good. So what I'd like to do is, and it's a small audience, and it's actually a lot of currently enrolled students. So that means you can get to do, we can have a quiz during the talk, so everyone has to stay awake. I'd like to cover kind of a very broad swath of human genetics and human medical genetics, both scene setting and using some examples, and then talk about a series of more vignettes or different examples of what's going on in human genetics. I'm not going to spend a lot of time on technology. The technology is amazing, and I want to just give you a very brief overview of what has happened technologically. But given the lecture series is about moving this to society and societal issues, I want to hint at where all of the technology is going to push the boundaries of these kind of issues related to public policy and science getting into our real life as opposed to just in the lab where I work most of the time. I want to start with a story as to kind of what and why we do this work. And I know if some of you in the back, it's a long room and a smaller screen. If you can't see, feel free to move up. I won't throw the silver dollar, the Canadian dollar at you. But on this slide is a image of the abdomen of a woman. And I don't know if there's a pointer, but I'll wander around. You can see that there's something different between this one and this one. Right? And so those of you in the front can see there's a circle on the slide that's in the same position in both images. And in the second image, the circle is empty. And so what's in the circle in the first image is a tumor. And it's essentially a metastasis from an ovarian cancer that was in this woman's abdomen. The second slide, which was taken a couple months later, was after she was treated with a drug, at the time and still is an experimental drug, but it was a drug that was designed and thought of based on the knowledge of genetics and cancer susceptibility. Not a drug that was thought up as a generic one to kill all cancers. And so in this audience, I bet I will get some hands. In older audiences, I don't, but I ask, is there anyone who doesn't know someone who's had chemotherapy? Right, so I do get a few hands. The rest of you know that the current treatment for cancer, because you've seen someone go through it, is not the ideal and not something we should be that satisfied with. And so some of the genetics advances, one of which in particular I've worked on a little bit, I can't take credit for this work, has led to these more intelligent design of drugs. There's going to be lots of stumbles and it's going to take a long time, and I think one of the themes of the lectures will be that we have a long way to go. We're moving very, very quickly, but we have a lot to do to get this stuff advances integrated into society and into medicine. But this is a great example of finding the gene. So gene was found in this case a long time ago before all the modern technology, but identifying a specific gene associated with the disease led to a design of a new drug that no one would have thought of without the knowledge of the genetics. And I'll talk about a few other examples. The promises of genetics and medicine is that it really will revolutionize medicine. So a lot of that's coming from my institute, which believes strongly in it. It also needs to constantly keep the excitement up. I happen to believe strongly in it that it will revolutionize medicine. I'm a little bit more circumspect about what the pace will be and when we will see some of the fruits. But along with that, since your genome and your genetic sequence is yours, there's the promise that you actually will be much more in control of your individual medical records and your individual genome going forward than maybe you are right now. And I know that most of you know that your medical records are kind of yours, but you don't carry them around, you don't have them with you for the most part. The medical system hangs on to them. During the talk, I think I'd like to cover these topics, and I'm also open to answering questions as we go along if something burning comes up, and I will stay afterwards to answer specific questions. But I'd like to spend a little time to get you thinking genetically, and I know some of you are biology majors, some of you are science majors, some of you are geneticists. I won't necessarily need this refresher, but it really is to, I'm doing very broad brush strokes to how to think genetically. I'll spend a minimal amount of time on the technology and the applications of the technology, and talk a lot about challenges and solutions on how we're going to go forward. And in the end, and I think I will have time, believe it or not, I'm going to talk about football. And football in a way that you probably wouldn't have predicted, in a way that I wouldn't have predicted a couple of years ago. And so why are we interested? Why are we pushing genetics? Why do you hear so much about genetics? Why do you hear advertisements that say it's in our corporate DNA? And this perfume is DNA I think is the name of a perfume. Why has that gotten into society so much? I think a lot of it is that there's the promise of prediction, right? So in the old days, prediction was in the realm of those that had magic. And in science, in a lot of the sciences, what we actually do is give ourselves the tools to make predictions. The physicists, the chemists do that exquisitely well. I mean, I know that if I throw this coin that I was given at a certain velocity with a certain angle, I can predict pretty reliably where it will land. And I can do it again and again with the same thing, it'll land in the same spot. Biology has been not in that realm for a lot of the areas because we're all complicated individual organisms. But if you think about it, genetics from the rediscovery of Mendel's laws has been a part of biology that is highly predictive. And I'll talk a little bit about what we knew 100 years ago and what we're trying to do now. So that's why genetics is getting a lot of hype, because it can be predictive. And if you can predict what's going on, in the terms of medicine, you can enhance prevention. We'd all like to prevent diseases. You can enhance early detection. So everyone knows if you can pick up something earlier, it's probably easier to treat and less likely to kill you. Diagnosis, I'm sure some of you have had people who have tried to find out what's wrong with them and spend a long time to figure out what's wrong with them. Genetics can aid diagnosis. Even if you can't pick up things early, genetics can actually help with prognosis. Is this tumor going to kill me? Which I think is a very important issue. Is this tumor going to be sensitive to X disease, which gets to the tailored therapy, X chemotherapy, the tailored therapy. So all of those areas, and I'm going to give a few examples of some of them, are where genetics will have an impact going forward, and some of them are, as I showed you on the first CT scan, are places where it's having an impact now. So let me just give you, and this, I'll read these for the people in the back who are too far away to see the screen, but let me just give you 3 cases, case studies, 3 quick case studies. So the first, and I made this slide before the PSA rulings that have been in the papers the last couple of days, but the first is a hypothetical case of someone who's now 60 years old. He was tested early on, maybe at college, who knows exactly when he was tested, and told that he was at increased risk for prostate cancer because of his genetic makeup, not for his PSA, just for his genetic makeup. So in his case, it made sense to screen for biomarkers. And notice I said biomarkers, not PSA, because PSA is somewhat of a sloppy marker. But think in the future, some kind of biomarker panel. And so that because he's selected for screening, those markers say that he is likely to have a lesion in his prostate. The medical community does a biopsy, and it confirms that he has disease, and then they look at the genome. They look at essentially the genetic makeup of that tumor material, and from that can predict that it's likely to metastasize and maybe kill him before something else does. And so they suggest intervention. The other thing that they can learn from that biopsy is what drug might be best treated if it's not surgically treated. There are elements of this that are futuristic. We don't know about biomarkers, but there are a lot of elements that we can do now. Here's the second case, and you can decide whether I'm getting more and more out there with these cases. A 25-year-old college student, graduate student, I guess, at 25, graduate student at 30. It's a long time for graduate school. She had her genome scanned at age 10, and you can think about issues of testing children about things that they don't have control over. But that scan could turn up that she has high risk of getting lung cancer. So at age 10, we know that she might be at high risk of lung cancer. And everyone knows what the other risk for lung cancer is, right? Smoking, right? In addition to that, she's found to carry 3 of the strongest predictors of whether or not she'll have an addictive personality. So if you wanted to target behavioral modification, this is a person you would really want to target. She has addictive tendencies based on her genome, and she has lung cancer susceptibility. So starting in junior high school, that information is given to her high school counselor or genetic counselor or something in the future that we call, and she is intensively educated about the risks of smoking more so than other students. And by age 25, she's through the window of where people experiment with tobacco, and she's probably never going to smoke. That sounds like kind of a good, it's a little scary when you get testing of children, but that sounds like not a bad use of this technology. We are, and just to tie it to reality, we are pretty far off on knowing the strongest of the lung cancer ones, and the addictive personality data is pretty weak at this point. But we probably can get those data. And then the third case, it's a childless couple. They want to have a kid. The dad is known to carry the breast cancer susceptibility mutation. They want to use in vitro fertilization because they need assistive reproduction, but they don't want to have a child that has his mutation. So they do in vitro fertilization. They take embryos and they test one cell of those embryos to see whether or not they contain the mutation. And then they implant into the woman only embryos that don't carry the BRCA1 mutation. And then they give birth to a child that does not carry the mutation. Does that one sound also futuristic and far-fetched as well? So some people are saying, yes, of the 3 cases, this is the only one that's been done so far. And so this has been done with existing technology in real-life couples. Now, it is not part of Medicare and not widespread. It's not part of mainstream medicine. But this is, of the 3 cases, 2 of them are much more futuristic, maybe in some ways more benign. This is the one that actually has been done and written up, and there have been couples who have availed themselves of this new technology. So some of this is here now, and others are coming in the future. I want to go back into just the basic principles of how we're getting to this point, and that requires reminding you a little bit about genetics, and then reminding you that this is not the genetics of today that we're going to talk about. So anyone who's not had Mendelian genetics, Mendel's P's, in a class sometime in the last 55 years? Right. You can get a pass. And so one of the reasons that genetics it has so much power with the public is because this is the public's view of genetics. And I said scientists who can predict things have this special power. And if you look, you can't see it, but those P's are different colors. And knowing the genetics of the P's, you could predict what frequencies the different colors will come out. And that's pretty amazing. It was totally astounding when Mendel did it, that he could predict it so radically. What you have to do to think genetics in the modern era is to go back also 100 years and think about what genetics can and can't predict. So this is a picture of identical twins. They're incredibly similar. Their genomes are identical, at least at birth. What's enhanced here is they're dressed alike, right? So of course they look identical. And is there anyone who's known identical twins? Anyone who's known them over a long period of time? So early on, could you tell them apart? Now that you've known them a decade or more, can you tell them apart? So some people are nodding yes. I used to live next to identical twins that are now in their mid-20s, late 20s. And as kids, I could not tell them apart. The other half of the equation is the environment. You can tell those identical twins apart because there are changes going on, even if they are still dressing alike. They're starting to look different. Right? So Mendelian genetics is very powerful, but it doesn't explain this. So for those of you that can't see it, and I don't know if did the lights go down one more notch, maybe? It's a picture of, and for those of you in the last several rows, or I was just going to do it by age cut, is there anyone who doesn't recognize these 2 guys? It'll usually be that's all right, you can raise your hand if you know. So it's Wilt Chamberlain and Willie Shoemaker, right? So in their respective fields, which I'll just give you a hint, one was a jockey, the other one was a basketball player. They were the best in the world. Does Mendelian genetics tell us why these 2 guys look different? Right? So that for those of you that think about genetics and crosses, if we cross those 2, would we get kids that were either Wilt Chamberlain or Willie Shoemaker? We wouldn't. We'd get something in between. And that's because the majority of human genetics and the stuff that we really are going to talk about today looks more like this. And so I hope you can see this. This is height. Right? So this is a selection of military recruits from about 100 years ago lined up in height order. Right? Who knows how strongly inherited the height is? Anyone? Very strong. Very strongly. So roughly 80% of height is inherited. Sounds very, very impressive. Who knows couples that that had kids who their height didn't seem to correspond to their height all that well? That's because even though height is strongly inherited, it's the product of a whole bunch of different genes. So we can't predict, like we can't predict Willie Shoemaker versus Wilt Chamberlain. We can't predict accurately who's going to be tall and who's not tall. Right? Height in the population is strongly genetic, yet follows this bell curve distribution. It's kind of a uniform distribution with different frequencies. The other thing that you should note, and again coming back to this theme, look at the peak of the bell curve. What do you think the mean and the median, for those of you that are statistically inclined, are in there? And this is in English units, not metric units. So I'd place it right about here, somewhere. And that's a little bit taller than me, but not much taller than me. Am I of average height or am I short? I can take it. You can. Right? You can say it. I'm short. For a guy, I'm short.
This was 50 years ago, too.
Right? So it was more than 50 years ago, and so why is that shifted? Nutrition, right? Nutrition. And so in this one slide, I'm illustrating a very strongly genetically determined trait that is widely modified by environmental exposure. Those are thinking genetically, especially for complex traits. This is the mindset you have to be in. We are not predicting the future with certainty the way we can with the Ps, and the way we can with strongly inherited diseases that you're familiar with, like sickle cell anemia or cystic fibrosis. A lot of the action and a lot of the movement and a lot of the data we're generating is to address more complicated issues. And it is to address the complex diseases that are kind of listed on here. So I could make the slide longer, but if you were to do away with the things listed on this slide, hospitals would be a lot more empty than they are now. And the population would generally be a lot healthier. And genetics contributes to all of these things, but it does so in combination with other factors. And these are kind of the factors that go into produce disease. They're genes, behavior, environment. There's also a random factor that no one really talks about, but there is a chance factor. Of those, we know that behavior, and I'm not going to talk about behavioral genetics or behavioral studies much at all today, but we know that behavior is probably the number one modifiable health determinant. Right? Genes are pretty important. It's where I spend all my time thinking, but I don't lose sight of the fact that they're only part of the pie. And obviously environment and everything that goes with it is important contributors too. And so human diseases and human genetics does not behave the way Ps do. But we spent the last 100 years working very strongly on the strong inherited disorders. Now we're tackling the ones that are harder to do. And we're doing so with these technologies that I want to spend a few minutes on. And the slide says small is powerful. So who are science majors in here? It's a lot of science majors. That's cool. Do you use test tubes in the lab? Right, so you use some test tubes. Note this is a wooden rack. Anyone use, this is a newer university. You still have wooden racks in the lab? Well, I can tell you that in the labs where you're doing small-scale studies, you still use these tools, right? And that's a rack that's about this big. And if you were doing one experiment and there's 2, 4, 6, 8, 10 spots there, right? So that's 10 samples you can analyze in that rack. What the technology has done in genetics is essentially gone very, very small on that. And so these are the kind of technologies used to generate the data. I'm just using an example, on the left side with all those dots, those are single little clusters of DNA molecules that are being sequenced on a microscope slide using what's called next generation DNA sequencing technology. That technology can sequence 30, 40, 60 million molecules in one experiment. So imagine test tubes, 30 million test tubes. But they've scaled down, and I forgot to bring one. I usually bring some of the slides. They've scaled down so we can do the entire experiment on a really tiny platform. The other platform, the thing to the right, again, is on microscope slides where we're doing genotyping, where we're just asking whether you have this variant or not. The slide there can ask, I think that has about 10 stripes. You can put in 10 individuals, different individuals' DNA on that slide and ask what their genotype is. What variant do they have at 1 million different spots on their genome? And you can do that in one experiment. The sequencing run, if I wanted to sequence one lane, which is millions of bases, runs at this point now just under $1,000. And these tests for looking at 1 million different genotypes, 1 million different spots on your genome run only a couple $100 per person. And so the technology has gotten incredibly powerful at producing lots of data. To give you a perspective, when I first started at NIH, we did a study where we were assaying in 5,000 people 3 different genotypes. So just 3 different spots on their genome we wanted to measure. And it took 2 people full-time a year to do that. We now can do that with a slightly different technology that's shown on here. One undergraduate student can do that in a week. And so we spend a lot more time looking at data and a lot less time generating data. And we spend less money doing it. Just to give you even more perspective, this is the cost of sequencing 1 million bases of DNA. So think of 1 million bases of DNA as a convenient unit, like pound of potatoes or 3-pound bag of potatoes. And again, for the science majors, you'll note this is a log scale, and the math majors will notice this is a log scale. For those of you that haven't had that in a while, a log scale jumps by 10 at each interval. The straight line shown on here is Moore's law for the technology-oriented, which is the cost of essentially transistor computing power. And that is what's given us all these fancy devices that everybody carries around that were unthinkable 35 years ago. And you'll see that the cost of sequencing followed Moore's law and then dropped off around here when these new technologies came available. And so to give you some perspective, 1 million bases has gone down from costing $9,000 to a penny or so. If during the course of the lecture my short height were to change that much, by the end of the lecture I would be 10 miles tall. And likewise, I recently got rid of my 1998 Honda Civic. If the price of the car from 98 till now changed that much, my new Honda Civic would have cost me $1.50. And so those are really amazing changes in data production. And this is what's generating a lot of the excitement and a lot of the buzz that you're hearing about in genetics, because we can get more and more data. Now, analyzing and dealing with it is a different issue, but we can get more and more data more quickly. I actually live in Baltimore, and some of you know the Hubble Space Telescope is there, and I had an occasion to talk to people from the Space Telescope. And we're thinking that, wow, those people have been dealing with tons of data for years. In biology, it's new to deal with so much data that you have to put it on portable terabyte drives. And so I talked to these people. I said, well, what do you must be up to 100 terabytes per experiment now. And it turns out that the Hubble Space Telescope data sets are now roughly of the same size as some of the genomics data sets. And I was hoping for some guidance on how to deal with these data sets. The first time we generated a whole genome association in the lab in Baltimore, we tried to send it down to the lab in Bethesda via the internet. And even though I think we get some preferential access, it turned out we used about 1/3 of the bandwidth on that one pipe between the two sites. And someone called up and said, what are you guys doing? That was a couple of years ago. We don't do that anymore because the infrastructure can't take it. We actually mail disks around. But biology is now generating data sets of the information content, not value, but just the total information content that are comparable to the kind of things astronomers are dealing with now. So what are we doing with all that great data? Here's a cover of Nature from a long time ago where it assumed that you could generate your entire genome, and you could use that to guide your life. And so how far along are we in that pursuit, and what do we need to do to get there? So I want to return to the slide that I showed you earlier and talk about a few examples and of each of these categories. I won't show slides, I'll just discuss a few of them. One of the ones that's the trickiest is prevention. As most of you know, that you can get genetically tested, and I'll show you some of the companies that do it now. If you have a credit card and an internet connection, you can get all of your genetics online after you send them some spit. The hope is that that's useful for prevention. Prevention is a hard thing to do, and I'll show you in a second. It's not really what mainstream medicine does very well, and some of you are probably more public health oriented. And I'll give you an example of the differences in mindset. I constantly come across them both going between both worlds of the clinical medicine versus public health type medicine. So that's one we need to do a lot of work on. The early detection is really in place and working now for some of the stronger susceptibilities. So for instance, the people who carry breast cancer susceptibility mutations in BRCA1 and 2, we know enough now that we can recommend different screening methods for people who carry those mutations. And there's been data to show that it actually can affect early detection of cancers. Diagnosis is one that I mentioned briefly, in that we're getting much, much better at understanding, especially in the field of oncology, what the genome of the tumors looks like and what that means. And that's a case where you actually go in and get a piece of the tumor and you analyze its genome, and you can compare that to the person's genome and make predictions. And I didn't bring a data slide, but they actually look, you can classify tumors into different categories. So one of the things that a colleague is working on is ovarian cancer, and so there's several different types of ovarian cancer. Right now they all get the same drugs, and about a third of the people, the drugs don't work, and they have to sit through a really awful course of chemotherapy. And so by getting bits of the tumor and looking at their genome, you can essentially maybe diagnose and then tailor the therapy toward the right drug. We need to do a lot of work on those, but there's already been successful stories with very simple genetic changes in tumors, most notably in lung cancer. For those of you that know the Gleevec story, where there's a specific rearrangement in the tumor that makes a new gene, and some very clever people said, well, gee, that gene that is formed by a fusion of two genes doesn't exist in the rest of the body, so if I make a drug against that one, maybe it'll just hit the tumor and not have those terrible side effects. And those are actually in clinical use right now, based on understanding the tumor. Prognosis is something that can easily be looked at by looking at a person's genetic makeup, correlating it with what happens to that person in large data sets, and then going back and saying, well, the last 100 people who had this profile went on to have disseminated disease or didn't need treatment, and therefore we're going to change the way we treat people. Because right now, even though physicians will say all medicine is personalized because we deal with the patient, it's not exactly personalized as much as we would like. And I think in the next example, I want to highlight this tension between public health and medicine. So we think that there probably will be advantages to knowing your genetic profile. I think most of you in the room probably can say, yeah, I think it should be useful at some point. And that's the medical genetics paradigm, knowing what the profile of the patient is before you. What about advantages of knowing everyone's profile? That's the public health paradigm, in that we apply things to the population. And they're not really that far apart, but here's two extremes. Does anyone know who that is? Any civil engineers in the room? No? So I spent time at Hopkins. That's the Hopkins faculty member that has saved more lives by far than any other Hopkins faculty member. Right, so it's Abel Woolman, who was a civil engineer.
Injury prevention?
Who figured out how to purify water, how to chlorinate water so it could be purified, and then went around the world spreading that technology. I used to see him when I was in graduate school. He lived nearby. That's a public health paradigm. Abel Woolman was moved by individuals, but he didn't treat individuals. He treated the entire population. So is that the opposite of what we do in medicine? Well, consider this example. Lipitor. You know, people think that's medicine, right? People, I won't ask if anyone in the room is on Lipitor, but given the sales, I'm sure there's some people in this room who are on Lipitor. This is an ad, so it wants you to take Lipitor. It says, reduces heart attack rate by 36% in the big print. Right? You probably can't see it, but there's an asterisk next to the 36%. And in the small print, which the FDA probably made them put on the advertisement, it says 3% of patients taking placebo had a heart attack compared to 2% of patients taking Lipitor. Right? So for those of you that aren't doing the math in your head, that means 100% of if the 100% of people took Lipitor, 97% of them didn't need to. Right? That is medicine in the public health model, because we're giving Lipitor to all of our patients that have a certain paradigm, and it's only decreasing the population by a little bit. It shouldn't be that far, but and I'm not criticizing this approach. It gets, statins get a lot of credit for decreasing heart attack risk. So, but the physician would be thinking he's doing personalized medicine, when he's really actually doing more public health medicine, one patient at a time. So what are the challenges? This is a statement that is now 3 years old. One of the challenges is training, right, and understanding. So I'll just read the text. Training of physicians and other clinical care providers to ensure that health delivery processes and standards appropriately incorporate knowledge of genetics and individual variation. That's the government saying we really need to do this. And that's the Department of Health and Human Services, which is my, and since you're recording this, that's my parent organization. I happen to agree with that. I don't agree with everything HHS says. But we have a lot of training needs, and that's because outside of the medical system, and for those of you who can't see it, this is just a selection of websites from direct-to-consumer genetic testing. Anyone in this room can go to some of these sites. I don't know if they're all, and the bottom left one, you can get your entire genome sequenced. And several of the others can look at 1 million different markers in your genome for very, very reasonable price. These companies offer some of the services that we use for research for less than we can do it in the lab. I haven't really thought about whether we could send samples from that to them and get the data. But that's outside the medical community. I think I'm not criticizing it because I think it's pushing innovation, it's pushing the technology. But it's what has to be done, what some of you in the room will do is figure out how to integrate these things into the medical system, because they're not integrated right now, and there's a lot of tension. Most of you would say, or some of you would say, yeah, I want to get my genome sequence, it's great. Knowing your genome is really important. And I just want to highlight on this slide a couple of other things that we launched into medicine as great because they make common sense and we really should do them. The first is find lung cancer early. So lung cancer is an incredibly serious disease, kills a lot of people in this country. So why don't we find it early by looking at people at risk and getting them chest x-rays. Second one is chemoprevention. Oxidation products are really bad for you. Why don't we flood you with beta-carotene or vitamin E, really good things for antioxidants. That's got to be great, right? Back pain and disc surgery is another thing that we do a lot of. Breast cancer treatment and bone marrow transplant, all of these things were employed and used extensively in medicine. And some of them still are. None of them work. In fact, some of them are harmful, and for those of you that saw the paper yesterday, vitamin E failed yet again to show any benefit. This was after years, and some of these, the autologous bone marrow transplants run about $100,000 each. No efficacy proven ever. No survival advantages. And so that's because we went forward with these things that made sense and didn't figure out till afterwards that what we were doing was the wrong way about it. The same tension and the same discussions are going about genomic risk profiles. I can take your DNA and I can tell you what your risks are for 50 different diseases right now. Should we do this? And there are two schools. Yes, we got to do it. It's now. It's driven by some people in the sciences and some people in the commercial companies that want to sell it. And then, surprisingly, a lot of geneticists saying, no, we can't do this. And there's a lot of fight going on about this. And I won't read this. You can read some of the different arguments on both sides. And some of the arguments are absolutely in the opposite direction based on the state of the art. It's almost like a political discussion as opposed to a reality discussion. And the project that Andrea mentioned, the multiplex project, was one we did at NIH to try to get some data as opposed to figuring out, I don't know what the answer is. I don't know whether we should be doing this stuff. But I don't want to make a decision based on no information. And that's where we are right now. So there are a couple different ways to resolve debates. In the old days, we used to duel. In politics, you just shout louder and longer than the other person. In some cases, you just change the rules. You have little kids who play games and they change the rules in midstream so they can win. You can attack the other side personally, which has happened even in these genetic debates. But the method that I would prefer going forward is to actually spend some money to design some experiments and collect data. And that's what I hope that the science majors will take home, is that there really is a lot of value in designing experiments and collecting data. as well as the public policy major. So how do we go forward? Well, we can do this the wrong way, and we're already starting to do it the wrong way, in that some of these commercial entities are going forward. They're not collecting data, or they're selectively collecting data. Right? So if you, obviously, if you go onto some of these commercial websites, there's success stories about how great their knowing your genetic profile was and how you lived longer and avoided diseases. Are they necessarily a credible source? They can be, and I'm not disparaging companies. They can do this really well, but they have an inherent conflict of interest in pleasing some of their investors. So we have to go forward scientifically. It has taken the NIH a while to start realizing that they have to fund those studies, and they are starting to put out what we call request for proposals in this area, as well as in the area of public policy to try to figure this out. I think some of them have been funded recently. But it wasn't the case 3 or 4 years ago. It's only starting now. I guess the government's slow to react. But I think the way forward really is to just bite the bullet, build studies, and try to generate data instead of figuring out afterwards. I want to finish with an example of a case where things just went forward without looking at the consequences. And that gets me to football. And I have to ask an embarrassing question. Do you have a football team?
No.
Okay, good. I didn't think so. And then I drove onto campus and I saw these banners for homecoming, and they had a mascot on there. I'm thinking, I know you have a really good chess team. But I didn't think you had a football team. So therefore, I'll use my alma mater's football team to use this. This is a story about collegiate football and genetics. And does anyone know that collegiate football and genetics are connected? So let me give you a brief story and just show you as an example of how we can plunge forward potentially without understanding everything and not necessarily get it wrong, but not necessarily learn anything either. And this has to do, rose out of this gentleman's case, unfortunately. So this is Dale Lloyd II. He was a defensive back at Rice University. And you'll see he was born in 1987. He died in 2006 at age 19 in pre-training conditioning drills at Rice University. After he died, it was learned that he is a carrier of sickle cell anemia. He's not the first, and he hasn't been the last, Division I college football player to die in training because he carried sickle cell anemia. He was, oh, sorry, let me give you, for those of you that aren't up to speed on sickle cell anemia, let me just give you a one slide, 2-point primer. So sickle cell anemia is a disease we've known at the molecular level since 1949. Even before we could sequence DNA, we knew what change in the protein caused sickle cell anemia. Sickle cell anemia is a disease, and it affects 1 out of 500 African Americans, 1 in 1,000 or so people of Hispanic origin. It also affects other people from Mediterranean countries as well. It's in a collection of a lot of hematology disorders. It is a disease. People who have sickle cell anemia almost always know they have sickle cell anemia, and it's diagnosed early. There's a state where you can carry, so sickle cell anemia, for those of you that remember genetics, is recessive. You need 2 bad copies to have the disease. You can be a carrier or heterozygous, have 1 bad copy and 1 good copy. And you'll see it, carriers are 1 in 12 African Americans. So in this country, there are millions of people who are carriers. They're completely fine. It is given the name sickle cell trait, which I think is a little bit unfortunate, and there were programs in the 70s for screening that really confused sickle cell trait with sickle cell anemia. Think, for the most part, under most conditions, sickle cell trait or being a carrier of sickle cell anemia is not a big deal. Right? For those of you that know even deeper or want to know more, this is a really cool example of the reason this particular variant is high in the population is because carriers are protected against malaria. And so it's one of the few cases where selection in human evolution actually has been pretty close to proven. So this is the landscape of sickle cell anemia. Dale Lloyd's parents thought that Rice ought to have taken better care of their kid And they sued Rice. And they won in 2008. But they also, in the settlement, got the NCAA to agree that they will screen athletes for sickle cell carrier status or sickle cell trait. And in April of 2010, after lots of discussion and a year delay, they made a law, and I've spent a lot more time understanding the NCAA than I've ever thought I would. It's a very interesting organization. But in Division I, they declared that if you're going to be in Division I and you're going to be a member of the NCAA, all athletes, student athletes, will be screened for sickle cell trait or show that they have been screened or sign a waiver that says the university is not liable for anything that happens to them related to that. And that was it. Then the NCAA said, just do it. Now, for those of you that have been listening about just launching into things, they just launched into this. And so last year, and every kid, student athlete in a Division I school, regardless of sport, ethnicity, or even risk, is being screened for sickle cell trait. And the NCAA punts this, no pun intended, to the universities. So that do you have a Division I team in anything besides chess? Swimming? Yes. Swimming? Any Division I? I won't ask your personal results. Any swimmers here? No? Have you been tested?
Soccer, they're all Division I.
Yeah. So at this university, I'm sure, unless you're non-compliant, there are athletes potentially in this room, and you don't have to raise your hand, who have been tested for sickle cell trait. And there are educational materials. They're kind of weak. But the real question is, does this make any sense to do? And did the NCAA spend time trying to figure out whether it makes any sense to do? The Divisions 2 and 3 are now looking at whether or not they should extend this to their athletes. So Division 1, as I found out, is over 100,000 athletes nationwide. If you add Division 2 and 3, the number goes up tremendously more. And so Vince Bonham in our institute and George Dover at Hopkins and I wrote an editorial kind of bringing this to the attention to the medical community and pointed out that it's the largest and most high-profile genetic screening program outside of newborn screening that has been launched essentially since the 70s without anything really in place to find out whether it makes sense and whether or not there's certain stigmatization. So for all of the testing, we can estimate how many people will be shown to be carriers. A large number of them will not be African American. African American students, athletes, their coaches will know whether they're carriers or not, and how will they treat them. Yep.
If they're carriers, they can't play.
I'm going to get to that. So no, they can play. And in fact, 1 in 12 are carriers. Right? So the NFL, you can predict how many carriers are now professional athletes in the National Football League. Right? It's a lot. They're playing, they're doing fine. In the meantime, and even before the Dale Lloyd event, there's been additional deaths of student athletes. Importantly, so the lower quote is from the Secretary's Advisory Committee on Genetics, and there have been 15 reports over the last 15 years of athletes dying who were sickle cell carriers. Of the 8 sickle cell carriers who died in the past 10 years, 4 of them occurred in students who the university knew they were carriers. That's kind of a red flag for building a program. If you've already identified people who are carriers and you're still losing them, then maybe the program itself is not what you need to do. It's interesting that Division 3 is the most academic and most looking into this, and I don't know whether they've passed it yet. I think they'll make the decision after the bowl season. But Division 3 said, we want to look into this a little bit more. And I want to highlight this comment here. They want to know whether significant differences in preseason conditioning standards in Division 3 obviate the need for mandatory testing. That goes back to the original point about genes and environment. Here's Division 3 saying, well, maybe we don't push our student athletes as hard and we don't have to worry about it. Doesn't that tell you something about what Division 1 is doing? I don't know. The divisions actually are very independently organized and regulated, but it says to me that potentially Division 1 ought to be looking at what's going on. And a lot of these deaths have been in what they call preseason training. They've mostly been football players or some track athletes. Preseason training happens when it's warm and hot. And dehydration and heat contribute to the adverse events of being a sickle cell carrier. And that's not news. That was known. In fact, if you think about the math, the military, which really pushes people to their limits, has had lots of sickle cell carriers. In fact, they had testing programs years ago, and they still, we got some information from the military, they still test people, but they don't modify the individual's training regimen. They ended up modifying everyone's regimen. So there's the public health model, maybe where you don't need to know the genetics. You just need to change the regimen that produces the adverse event. So I want to close with going back to this when you're thinking about genetics, especially the new genetics, the complex genetics, and thinking about all of these components. So at the face of it, the NCAA program sounds great. You'll identify these kids and you'll prevent any kids from dying. Sounds great, but you have to show it actually works. And you have to show that what is happening to the rest of the people who have this condition don't have any adverse events. And to answer your question, there is no prohibition about being on the team if you're a carrier.
And there's no way to tell if you're at risk?
There is I mean, you are at risk if you're a carrier and you're doing preseason drills in the heat without enough water.
Okay. But there's no way to tease out a smaller
portion of the carriers. I don't know the answer to that. It would be interesting to.
What about the false positive and false negative ratios of the test?
So the test, and I can tell you that you've all heard about student athletes dying and professional athletes dying. The majority of them, or at least a large number of them, aren't dying because of sickle cell. They're dying because of cardiac arrhythmias. Several of them are inherited, but that test is very expensive and cumbersome and hard to do. In fact, I've talked to several Division I athletes. They don't even get EKGs. The test itself for sickle cell has been around for 30 years. We actually, when I was looking into this, we actually bought one and ran it in the lab. It's a very robust test, meaning that it has a very low false positive rate and a very low false negative rate. There are a few other hemoglobin conditions that can give you a false positive that are generally picked up by a second-tier test. The universities were left to administer the testing. The basic test is a $6 to $10 test. It's very easy to do. I don't know exactly how the universities are doing it. There's some of them, they do the first test. If it's positive, they do the follow-up test. And together, those two tests have a very, very acceptable false positive, false negative rate. So it's not that the technology here is actually pretty good. It's actually old technology, but it works pretty well. So I just want to close with where I think we stand. I think if you look at this, whatever that is, little kid crawling up the DNA helix, I think we're learning how to use DNA. I don't think we're the masters of learning how to use DNA and genetics to change society yet. I think, and this is my quote, it's not a quote from them, I think that almost every important question related to using genetics to improve health has yet to be answered. So for those of you that are in school, stay in school and answer them for me. And the harder questions aren't in what we do in the lab. The questions about, does this technology work? Can we make it better, cheaper, faster? Those are the easier questions. The harder questions are, does it make sense to do it? Is it cost effective? Do we have a professional base that can actually employ them? Is there a social acceptance for this? Will contribute to health disparities. What are the legal frameworks for knowing this? There's a lot of privacy and protection issues that this work brings up. These are the really difficult ones, and why I think talking to an audience that comes to this series I think is important, because there's a lot of opportunities for those of you that are thinking about what to look at in a research setting who don't want to be going into a lab to do DNA testing. can answer these questions related to DNA testing, which a lot of the people creating the test don't have the expertise to do. And so I just want to close with the mining algorithm again, and that we really are at the early stages of prospecting. And I think some of the medicine based on genetics is going to be major gold, and others still probably are at the stage where we don't know whether they're real gold or fool's gold. And the way we have to do it is actually just testing them rigorously. And so I'm happy to answer questions. Thank you for being very attentive. Go ahead.